Today, we will discuss signal transduction. In particular, we will be looking at GPCR signaling, including its importance as well as key roles it plays. Before we get into the details of GPCR, it is important to not only understand what it means and its function, but also to understand the history and its significance. The discussion will cover classification, as well as the physiological roles it plays. We will also talk about structure and how to look into how structure dictates function. After understanding the mechanisms that come into play, we will look at signaling. Two examples of pathways involved will be explored to help better understand the role of GPCR. We will conclude today's discussion with receptor regulation. G-protein coupled receptors, GPCR, are also sometimes referred to as 7-TM receptors, G-protein linked receptors, or GPLR, or also serpentine receptors. They are considered not only the biggest, but also most diverse group of membrane receptors found in eukaryotes. All animals express hundreds of different G-protein coupled receptors, GPCRs, that are responsible for responding to outside stimuli. A great way to view GPCRs is to consider them a mailbox that receives messages. The method of these messages can vary from sources such as light energy, peptides, lipids, sugars, and proteins. This information can have a multitude of meanings, anywhere from providing information as to what nutrients are available in the environment to information that is being sent from other cells. This means that GPCR-driven signaling pathways are in some way involved in almost every physiological function. In addition, they are in many of the pathologies present. This provides a small glimpse into the significance and importance of GPCRs and why it is important to understand the mechanisms that come into play. Playing such a vital role in an array of functions within the human body, GPCR understanding has had a tremendous impact on modern medicine. So much so that it is believed that 1/3 to 1/2 of all marketed drugs work by binding to GPCRs. This translates to roughly a global sale of $180 billion in 2018 alone. It is important to understand that much of this has happened in a fairly short period of time, within a little over the past 2 decades to be specific. In 2000, the first breakthrough was the crystal structure of GPCR rhodospin. Later, when the crystallization of the beta-2 adrenergic receptor was discovered, it revealed a few stark differences from the previously discovered crystalline structure. This was a ligand-binding receptor, and therefore the structure was quite different, but relevant since it would be the groundwork upon which modern medicine developed drugs. In 2012, a Nobel Prize was awarded for the research on understanding how G-protein coupled receptors function. The importance of GPCR is so great that research and discoveries have continued to be acknowledged. In fact, 7 additional times by the Royal Swedish Academy of Sciences that awards the Nobel Prize. Using genome sequence analysis, it is believed that there are over 800 GPCRs, of which 350 are for detecting hormones and growth factors. It is also believed that roughly 500 serve as receptors for olfactory and taste sensors. There are 150 or so GPCRs that are found in the human genome for which we don't know the function. In almost 25% of all cancers in humans, there is a mutation of the RAS, which leaves a very weak GTP binding site that essentially eliminates GTPase activity. There's been some debate as to how to properly classify the superfamily of GPCRs. All GPCRs have a common structure and mechanism of signal transduction. Originally, the superfamily was divided into 3 classes that were termed A, B, and C. Of these, the majority of the GPCRs were in Class A. Since then, there have been revisions to the classification format, and now the more widely accepted structure includes classifying the superfamily into 6 classes, either by letter or number. The classes are as follows. A, or 1, rhodospin-like. B, or 2, secretin receptor family. C, or 3, metabotrophic glutamate pheromones. D, or 4, fungal maiden pheromones receptors. E, or 5, cyclic AMP receptors. And F, or 6, frizzle-smoothened. Understanding the classification groups helps us better understand the roles GPCRs play. As already briefly mentioned, GPCRs are involved in a number of physiological functions. In the introduction, we had mentioned how GPCRs can respond to various stimuli, including light energy. 3 senses that are aided through the presence of GPCRs are vision, smell, and taste. The opsins are proteins that are light responsive and can interpret electromagnetic radiation into signals playing a vital role physiologically in vision. The sense of smell is aided through the olfactory receptors that bind odorants as well as pheromones through the vomeronasal receptors. Another sense that benefits from GPCRs is that of taste. The GPCRs located in taste cells can regulate gustduction as a response to bitter and sweet tasting substances. There are a number of neurotransmitters that are bound that play a role in behavioral mood regulation. Some of these include serotonin, dopamine, histamine, and glutamate. GPCRs play a role in the regulation of immune system responses and inflammation. For example, histamine receptors can bind with inflammatory mediators to target cells. This is often utilized for the inflammatory response. The nervous system also benefits from GPCRs. Both the sympathetic and parasympathetic nervous systems are regulated by GPCR pathways. Some examples include digestive processes and heart rate. Additional physiological roles include sensing cell density and metastasis of cells in tumors. GPCRs also help regulate the body's water balance to maintain a level of homeostasis. As we will discuss later, GPCRs are also involved in the endocrine system, playing a vital role in the CAMP signal pathway. There are 2 main types of membrane proteins, integral and peripheral. Integral membrane proteins are permanently attached to the membrane. This is important in understanding the receptor structure of GPCRs. GPCRs are integral proteins that are composed of 7 transmembrane helices, TM for short. This polypeptide that is embedded into the plasma membrane consists of 7 segments that span the entire width of the membrane. Thus, GPCRs are also called 7 transmembrane receptors. Structurally, the cores are very similar. As stated, there are the alpha helixes, TM, intracellular C-terminus, and an extracellular N-terminus. Being integral proteins, GPCRs are usually confined on the membrane. They have a ligand-binding element that is exposed on the extracellular side of the membrane. As mentioned, most of the receptors belong to the class A that have a pocket for ligand binding between the helices. The class B receptors have a large N-terminus that interacts with peptides since it has a greater attraction ligand binding site when compared to the pocket inside the helices. There are also structures that include 2 7-TM units. There are class C GPCRs that bind with allosteric modulators so that after the deletion, they can function in a similar manner to that of a class A receptor. GPCR proteins are what are considered heterotrimeric. This means that they have 3 components comprising their G proteins. There are 2 components that are attached to the membrane by lipid anchors, which are the alpha and gamma subunits. The 3rd subunit is known as the beta subunit. A comprehension of the receptor structure helps one better recognize the relationships between structure and function. As discussed, the GPCRs consist of an extracellular N-terminus, which is followed by the 7 transmembrane alpha helices. These are connected by 3 extracellular loops and 3 intracellular loops, as well as an intracellular C-terminus. The GPCR takes on the shape of a channel or tube with the 7 TMs forming a cavity within the membrane, which will serve as a ligand binding site. Larger ligands that can't fit within the channel may bind with the extracellular loops located on the N-terminal tail. In some GPCRs, the N-terminus can go through a conformational change after glutamate binding to an mGluR. Aside from ligand binding, the N and C-terminus serve other functions. The C-terminus usually composes of serine or 309 residues that phosphorylate. This in turn will increase the intracellular proteins termed beta-arrestins. Once the beta-arrestins bind, they lead to the enlistment of other proteins that end up creating signaling complexes. These complexes include the extracellular signal-regulated kinase, or ERK for short pathway, that activates endocytosis. GPCRs relay a number of extracellular signals intracellularly through the coupling with G proteins and arrestins. Lipid rafts are microdomains of the membrane that are cholesterol and sphingolipid abundant. A covalent alteration of cysteine residues leads to targeting the receptor of the lipid rafts. This is termed palmitolylation, where one or more of the intracellular loops or the C-terminus are involved in the modification of the cysteine. All G proteins use the same mechanism for switching between an inactive conformation and an active conformation. GPCRs are activated from an extracellular source known as a signal mediator or ligand. Once this external mediator interacts with the GPCR, it initiates a conformational change in the receptor. This change is what is then termed the activation of the G protein. The protein is then later inactivated by regulators of G protein signaling, or RGS for short. These function through GTPase activity. There are primarily 4 key mechanisms that are utilized by GPCRs. The first of these is ligand binding. In this mechanism, there's a receptor or multiple receptors for a multitude of ligands. There are what are known as orphan receptors, which are receptors for which we have not yet identified. Examples of ligand receptors can include sensory signal mediators, as discussed earlier, that are involved in aiding our senses, as well as members of the vasoactive intestinal peptide family. Biogenic amines such as dopamine, epinephrine, and histamine are also great examples. In addition, there are molecules known as chemokines and lipid mediators, as well as peptide hormones. This vast diversity gives an inclination as to how varied the signal mediators can be. There are also conformational change mechanisms that take place. In this case, in the inactive form, the GPCR is constrained to a heterometric G protein complex. There are 3 types of ligands that may be present. The first is a ligand that can push the homeostatic balance towards an active state. These are called agonists. Conversely, there are ligands that can move the balance towards an inactive state, which are termed inverse agonists. The third type of ligands are neutral antagonists that don't change the balance in either direction. When an agonist binds to the GPCR, it initiates a conformational change that is transmitted to the alpha subunit. At this point, the alpha subunit will swap out guanosine 5' triphosphate, GTP for short, for guanosine diphosphate, GDP for short. This shift instructs the release of the alpha subunit and the beta-gamma dimer subunits, as well as as well from the receptor. The dimer, consistent of the beta and gamma subunit, as well as the alpha subunit, are now free to interact with intracellular proteins by diffusing laterally on the membrane, which will continue the signal transduction. Also at this point, the GPCR is now free to form a new complex and start the cycle again of signal transduction. When the GTP is hydrolyzed back into GDP, the subunits will reset to an inactive state and the G protein will find an inactive GPCR. This process illustrates how the G proteins function much like a control that can be turned off or on by the signal receptors found on the cell surface. G proteins in an inactive state begin as a trimer of the alpha, beta, and gamma subunits when bound to the GDP. When a receptor activates, it allosterically activates the G protein. This is accomplished through the exchange of GDP for GTP. This exchange is ensured by the cell through maintaining a greater ratio of GTP to GDP. The units released from the receptor essentially created an alpha GTP monomer and the previously mentioned dimer. These two components will now diffuse along the intracellular side of the membrane, helping regulate intracellular proteins. Some of the targets of the activated G proteins include enzymes that go on to produce secondary messengers. Some also allow ion channels to act as secondary messengers in the interactions with ions. To control this activity, while some initiate the relationship, there are also those that function as inhibitors. Since the alpha subunit has a weak GTP to GDP hydrolysis capacity, eventually the inactive form will regenerate. This will then also begin the reintroduction of the beta-gamma dimer to a GPCR that is ready for activation. RGSs can control the rate of hydrolysis, which in turn can be accelerated when required. There's also evidence that illustrates crosstalk of GPCR signals that interact downstream with integrin signals. A great example is that between calcium ions and the CAMP signaling systems. The enzymes adenylyl cyclase and phosphodiesterase are stimulated by calcium ions. Changes in localized calcium ion levels can therefore have a direct impact on CAMP, allowing a cell to respond to more than one signal with extremely fine metabolic fluctuations that are localized. GPCRs utilize two primary methods of signaling transducers, G proteins and beta arrestins. G protein coupled receptors are the largest family of signaling proteins. The beta arrestins have a greater attraction to phosphorylated GPCRs, therefore leaving most of the signaling through to the G protein activation. As we will discuss, there's still the chance of interaction that is facilitated through G protein independent signaling. G protein dependent signaling involves the activation of 4 different types of alpha subunits present. These are all classified based on their sequence homology. Most GPCRs have the capacity to activate more than one type of alpha subunit. However, there does appear to be a preference for one type over the others when possible. The binding to the alpha subunit is dependent on the ligand involved. This is known as agonistic directed trafficking or sometimes functional selectivity. It is important to understand that while this might activate an alpha subunit, that their action could also activate other types of alpha subunits that are not the preferred target. A great example is through phosphorylation, where the receptor modification calls for a change in the G protein choice. The two main effectors of the 4 alpha subunits are cyclic adenosine monophosphate, CAMP, and phospholipase, C-beta, PLC-beta for short. In humans, CAMP is crucial in the body's response to hormones, nerve transmission, and sensory input. In the case of CAMP, it generates the enzyme adenylate cyclase, AC for short. The enzyme is responsible for catalyzing the conversion of adenosine triphosphate, ATP, to cAMP. While interaction with one type of alpha subunit stimulates the G protein, there's also the interaction with another alpha subunit that inhibits AC from generating cAMP. Cytosolic levels of cAMP can decide the activity in numerous ion channels, as well as protein kinase A, pKa. In that pathway, cAMP is considered what is called a secondary messenger, and the pKa are secondary effectors. Phospholipase C is an enzyme that synthesizes two secondary messengers. These are diacylglycerol, DAG for short, and inositol 1,4,5-triphosphate, IP3 for short. The DAGs will activate protein kinase C, PKCs, whilst the IP3 receptors will release calcium ions from the endoplasmic reticulum. This pathway is key for many of the body's processes. One example is the thrombone receptors in platelets that utilize this pathway to help initiate blood clotting when required. Since some PKCs are also activated by calcium ions, it is safe to assume that both pathways can congregate onto one another to signal the same secondary effector. As stated, while GPCRs are predominantly thought to work together, there are few instances where a G protein independent mechanism takes place. This signaling takes place through beta-arrestines and GRKs. While not common, this type of signaling has a great deal of physiological importance. The chemokine receptor CXCR3 plays a central role in the inflammation through T cell migration in a number of diseases. The chemokine receptors can display a predisposed agonism through beta-arrestin mediated signaling after receptor binding. Another example is that of the activation of the bradykinin B2 receptor, which inhibits cell proliferation. The relation between the GPCR and the tyrosine phosphatase SHP2 creates an anti-mitogenic effect of the bradykinin found in kidney cells. There's even research that alludes to the possibility that heterotrimeric G proteins might participate in non-GPCR signaling. We will now discuss the significance of the cAMP and phosphatidylinosyl signal pathways. Cyclic AMP can also act as a second messenger for a multitude of regulatory molecules. These regulatory molecules include hormones, growth factors, and other molecules that change the intracellular cAMP and therefore the associated pKa activity. An increase in cAMP stimulates the pKa to phosphorylate and activate two key proteins. These proteins are critical to the deployment of fatty acids of accumulated fats. The anterior pituitary produces a hormone called adrenocorticotropic, which binds to receptors in the adrenal cortex, activating the glycoprotein adenyl cyclase, increasing cAMP levels. Again, this makes the pKa phosphorylase activating several enzymes involved in the synthesis of steroid hormones. Some hormones will inhibit adenyl cyclase, lowering the concentration of cAMP and thus curbing the phosphorylation. One such example takes place in the pancreas, where the binding of somatostatin to its receptor leads to the activation of a G protein that inhibits adenyl cyclase. Extracellular signals like epinephrine can have different outcomes on different cell types. Three factors that contribute to the outcome are the receptor in the tissue, the type of G protein involved, and the pKa target enzymes found within the cell. Phosphatidylinosyl signal pathways, as alluded to earlier, involve IP3 and DAG. The conversion of extracellular signals into intracellular signals is also called a double messenger system. These signaling pathways mediate cell proliferation, survival, and metabolism. In addition, there are other cell activities that are mediated, such as membrane dynamics, endocytosis, and cell migration, to name a few. GPCRs can become numbed if they're in contact with their ligands for extended periods of time. This takes place in two different forms. The first is what is termed homologous desensitization, where a receptor decreases its response to an agonist at an elevated concentration. This happens through an uncoupling of the receptor from the signaling cascade after persistent contact. Essentially, the activated GPCR is downregulated. The second form is termed heterologous desensitization. It is also referred to as cross-desensitization, where unresponsive cells occur that would normally be responsive to an agonist. The receptor is free of its ligand and returns to a state where it may begin to respond again to ligands and begin the signaling pathway process again. Today we have covered a number of topics related to signal transduction, specifically GPCRs. While we have covered their history and the significance of their presence, we have also explored their structure to better understand the mechanisms they employ. Only a few of the pathways were touched upon, revealing the great complexity and numerous physiological influences GPCRs have. I would encourage everyone to follow up this discussion with two questions that would foster further discussion. To understand the importance of GPCRs, consider that selective regulation of GRKs and arrestins demonstrates the possibility to be used as a therapeutic resource. References have been provided for any further research you may have. Thank you.